Okay.
So, hello, Egon. We are now online. And yeah, hello, everybody, and welcome to today's Physical Science in NFDI Colloquium. This event is a collaboration between several NFDI consortia to showcase good data management practices to an international and interdisciplinary audience. I'm Dr. Sara Espinosa, and I will be moderating today's session. If you have any questions during the presentation, please use the Q&A feature below. If you want to ask your presentation directly, please raise your hand and wait until the end of the presentation. Keep in mind that this event is being recorded and it will be uploaded to our YouTube channel at NFDI BiGCaT. So we are pleased to welcome Dr. Egon Willighagen. Egon is an assistant professor in the Department of Bioinformatics, BiGCaT at Maastricht University. So I will not go into his remarkable professional achievements today, but rather tell you that the reason I cannot think of a better speaker for this colloquium than him is his passion for open science. While other researchers are wary of open science and fairness, Egon lives these principles and is here today to tell us about his experiences and his journey in the open science chemistry world. So without any further ado, Egon, thank you so much, and the stage or the screen is yours.
Yeah, thank you so much. I will share my slides now. It's a great pleasure to be here to share my ideas about open science and fair knowledge and data dissemination. And I will give an overview of what this journey has been for me, and some of the things that I learned, some of the things that we're trying to do, and we here as things that we do in our research group, but very often in collaboration with many other people, as an open scientist would like to see. So I will give some sketches of what we have been doing in chemistry. I will take the angle a bit from toxicology, because my recent research funding is mostly in this area. So the more interesting things of recent events are from that area, and that is reflected here on the inset. So the slide deck that I'm using here has been part of other presentations as well. And but there are some aspects here that I didn't mention before. So what I would like to start with is just something that really matters to us a lot, to our research field, but also is a very important discussion around open science and science in general as well. For a journal article, the sustainability is pretty simple. The journal stays around normally. Not always, but in most of the cases, there is always a library that has a copy of an issue, of a journal issue. For data and software, this is more complicated. First of all, it's not printed on paper or something, it's digital, so it requires something to run it. So the question of sustainability is something that is very important. The longer term availability, however, has another thing. So where journals will be sent to libraries, this is also different for software and data. And this is one reason why I got interested in open science. So some 20 years ago, 25 years ago, when I was a student, I ran into the problem that I did not have access to software I wanted to look at to see how they did it. I wanted to look at the methods section, but something like a methods section is pretty straightforward for an article, but not so much for data or for software. So you need really access to the source code. Now, access to data and source code has been a topic of interest for much longer than the period that I've been doing open science, and actually since we've been calling things open science, which is something well close to 25 years now. But yeah, there too you see evolution, and we'll see part of that. But sustainability is an important thing here, and I think open science helps here. The concept of open licenses help here because they allow other people to pick up things from before. So mathematical formula, for example, is very easy to reuse. There is no copyright on a mathematical formula, but for a lot of other things, this is quite different. And yeah, as an example here, how this concept has helped us a lot in toxicology, in computational toxicology. A nice example here is something that was started by the OpenTox project, and I was not involved in that project other than being an user of it. And later on, I gave feedback to them. It was a pretty open project, but they also were open science from the ground up. And so their database, the OpenTox database software, the reference implementation of the API, the open standard that they developed, that actually got reused in the next project in ToxBank. And I was in that project involved as a postdoc for 1 year, and then just before I moved back to the Netherlands. And back in the Netherlands, I had the chance to start the eNanoMapper project, and there too we wanted to use the open approaches. We got the funding from the European Commission, and we continued with the OpenTox software. And in the eNanoMapper project, so that was in the field of nanosafety, of nanotoxicology, we made a convincing point apparently, because since then a lot of other projects actually have adopted the open solutions that we continued there. So this diagram that you see here from left to right about various projects, we really see this growth, this growing adoption of the open approaches. And with the notion that not all of those projects, they're still involving all of the original developers of the platforms. Though in this case, there is one company, ID Consult from Nina Eliaskova, that plays in this case a very specific role, but it is all open source. So that allows other projects like PHP for Safety, where she is not involved, or Open RISNet to adopt these approaches as well. So so that is about sustainability. So for sustainability, we need to be able to continue building on something from the past. So basically, the standing on the shoulders of, and what we need to stand on in my field of research, so computational biology, computational chemistry, are things that involve open data, open standards, open source, open repositories, a very piece of infrastructure. We need to be able to repeat things, so be able to rerun an analysis, which is particularly interesting for computation. And there, the rerunning can have different levels. So either you want to repeat the exact same calculation, or perhaps you want to rerun an analysis, but with new data. So the old data plus data that became available in the last year since you last ran the computation. And another important thing here at the bottom here is the peer review. So one important aspect of open science is that starting from something that we know from open source, the release soon, release often paradigm, the idea that when you share things early, people can actually look at that. And just like how peer review helps us write journal articles, getting the details of the experiments right, getting the argumentation right, getting the details correct and sorts, the same applies for data and for source code and for standards. And so the advantage here that I see is that with this release soon, you actually give people earlier the chance to review it. And the most obvious example here is people just reusing it. And in reusing it, they do an in-depth review of what is going on. Yeah, I've seen a lot of positive results out of that early peer review, so where we have been using this is in things like Smiles, which for a very long time did not have a specific open license, but it was openly available. There's now Open Smiles. There's Chemical Markup Language, which will say a bit about. There are standards like the nano, InChI, and Bioschemas. The last one I also show a bit about. And then the open source and open source Bioinformatics. I'll show some results about where I've been quite active, but there is a lot of open source software nowadays, very commonly referred to as research software. And this too, not all research software is open, of course, but there's a whole field of new kind of science where a lot of things matter, where the peer review is essential, because source code is algorithms. The algorithms have to be implemented. This has to be done correctly. And this translation from an algorithm, from the standards, from the ontologies that are included in the software, and the translation of that to an implementation, you cannot review that based on a journal article. I believe. So really access to that source code is important there. Now this is possible without open licenses, and in chemistry this is not uncommon actually to have access to source code without having the ability to modify it. But there are obviously benefits for that as well. And that brings me to this slide actually. There is open science, there is fair, and there is being used. And these things are actually three different things. There is some overlap, but they're definitely not the same. For fair, we very often think of this as part of open science, but fair is not open. They are very strong together, but they're separate things. You can be fair without being open. But the bottom thing here, the third thing here of being used is also independent. So where fair is about reusability, is about being reusable, it doesn't necessarily go beyond the theoretical. If you don't have access, if you're not, even if it's as silly as you're not rich enough to get the access, then you will not be able to reuse it. But there are a lot of things where it can be fair, but still not reusable. But the same thing we see for open. If I look at my experience as an editor-in-chief of a Bioinformatics journal, it's not uncommon that people make things available under an open license, but it is more like a dump on the community. They haven't been working on this release soon, release often, and it is open, but it's still not really reusable. So these are three different aspects that are closely related, but in my experience, it's very useful to think of these as different aspects, and because that makes it easier to think about the problems and the solutions for the appropriate situation. So that was a bit of introduction. That will make the rest of the slides a bit more, give it a bit more context of why I'm saying this. So this was the abstract that I wrote up for this talk, and I will go through a number of these things. I started this talk with sustainability and the difference between fair and open. And there is something here that is important, and that I want. Oh, there's a slide missing here that I'd like you to focus on. It's probably not missing. It's probably further in the slide deck. But in this abstract, in the middle there is something like made interoperable with community languages and explicit semantics, and reuse according to the best domain practices. And this is the hardest part, and this is also the most interesting part. Making different data linked to knowledge, different knowledge bases, possibly at a different level of detail, or a different level of the information that it captures. Different models for that perspective. For example, gravitation versus a macroscopic property compared to quantum mechanics. different models that are hard to combine, but at some point you still want to combine that in some way. And that is where I think the interesting science comes in. So the interoperability and the actual reuse, that actually in the end determines the sustainability. But we should not distract ourselves too much with trying to figure out how to do it, but actually do things. And that gets us back to the release soon, release often. Just as a quick reminder here, but I assume that everyone knows about findable, accessible, interoperable, and reusable in this this context. So I will not say much about that, but just give you a second here. The R1.3, that is the community standards. That is one particular thing that we have been looking into. So how do we translate our community norms on how to do science, what quality science is, the representation, how do we translate that into something that we can work with in this, this context? And that was triggered actually by an earlier study we did a couple of years ago, where we looked into how fair are the nano-safety resources and some of the other resources. And then we basically observed that finding findability and accessibility, that's a lot easier than actual interoperable and reusable. And you can see here that the R1.3 at the far right here, that typically scores very badly. And that is that has to do with that distraction. We've been thinking a lot about, well, if only we make our data or our software or our standards openly available on the web, we just link it to an open license and then we're done. And that is not the case. And that should not be the goal. It's a step that you probably want to take, but it's definitely not the goal. This was the slide that actually wanted to show. So instead of instead of focusing on the F and the A, the interoperable and reusable, that has been my interest long before FAIR existed, and we'll see a bit about that. And the recurrent topics in this presentation that will come back all the time are things like that support this this interoperability and the reuse, and those are things like identifiers and controlled vocabularies or even ontologies, the use of semantic web technologies like resource description framework, we'll see of that, and a number of other common standards that help us link things together and allow us to do data analysis across two or more different sources of information and data. So the first thing that I'm going to show is something about open science research outputs that we have been doing is the ontology. Or ontologies, I have to say. And one of the ontologies that we have been using and to link different sources together, bits of information and knowledge graphs together is the chemical information ontology. And this is a project, well, 12 years old already, where we looked into how can we represent chemical information. And one important, I think, or one very useful, I have to say, perhaps a distinction there is that we looked at things that, the notion that we have experimental data, but also that we have computer data. And that experimental data might derive to derived data where software was involved. Think, for example, here about crystal structures. If you use X-ray diffraction, you have your raw data, but you don't actually use that. You use the crystal structures that you derive from that. But there is software in between, and that software implements a certain algorithm, and that algorithm can calculate a descriptor. So in that sense, the descriptor here, something that describes the crystal structure, would be the crystal structure that we would download from the PDB database or from the CSD database or the iCSD. That would be a descriptor of the original experimental data there under this model. In cheminformatics, there is a lot more of that, because there we're very much used to things like a SMILES and a chemical markup language where we describe the chemical graph, but even the chemical graph is an abstraction of the chemistry that is underneath. The physics of chemicals is a lot more interesting than just the graph. More recently, the eNanoMapper ontology was created. That was done in the eNanoMapper project that I mentioned before. So that project had a database site and an ontology site. And the ontology here to get the community to actually speak the same language. That was what the European Commission asked for, and we opted here to use an ontology. What you can see in this diagram is that we're actually reusing a lot of other ontologies. So we're not doing things from scratch, but we created an application ontology where we used the bits and pieces that we liked from other ontologies to capture, to allow us to integrate data from different research projects, from different providers, and sometimes also with the whole different research fields. So the more biological side with bioassays from the, which we see here in the middle of that diagram there, the bioassay ontology, BAO. And on the other side, things like environmental aspects in the ENVO library, or the chemistry, the ChemInf ontology, and the nanoparticle ontology and so on. So integrating data by integrating different representations of the knowledge. And this ontology, along with the database, is also still used. So the sustainability of that actually turns out to be maintainable. Let me phrase it like that. We definitely not solved the problem, but we created something that we can still use. For the cheminformatics, we see a similar thing. So I realized when I was preparing the slides for this talk that this slide that I showed for the eNanoMapper, starting from open talks, are now widely used. I could and maybe should have done that for the chemistry development kit as well. The CDK was founded in 2000, actually. We had a meeting where Chris Steinbeck, he's in the NDFI for chemistry, and then Gazelta from JMOL. We had a meeting and we realized that JMOL and JChemPaint they were both cheminformatics tools, and they were both written in Java, could be combined. And in that meeting in South Bend, in Notre Dame University, where Dan is located, we thought of starting to develop the chemistry development kit. So this toolkit is now 23 years old. The code base actually goes back as far as 97. So quite before that, when the CompChem libraries got their first bits of code, at least. And I think JMOL started around the same time, originally separately. So here you have the two tools. The top left is the original Java JMOL version, and bottom right is JSMOL, the JavaScript version of JMOL, but still based on the same code base. These are open tools where there has not been a significant amount of funding, but it was sustainable by this notion that, well, we can pull together our small bits of resources and can take care of this library over time by multiple people. So Dan Gazelta was the original author of JMOL. It has a couple of people in between that were responsible for making the releases, for answering the questions, and for the last, at least 10 years, Professor Bob Hansen has been this Doctor Who of JMOL, making sure it keeps running in the current environment. The other thing of the open licenses, they're very often linked to, they're not expensive, or they are actually even free. We see that with a lot of open source, that you basically do not pay for that. Those things are not implied by the license, but it is the wish from the people doing it. And so that allows us, for example, so this is work from one of my PhD candidates, Amar. And he actually integrated a lot of docking related software because he had an interesting thought. He was wondering how would the binding affinity of small ligands, and ligand here in green being bound to a protein, how does that binding affinity actually change when the protein changes because of single nucleotide polymorphism? And so he started combining different sources of information. He started looking at biological databases and look at, well, what are the different forms of proteins that are abundant around the world? Which SNPs are prevalent in different populations? And he selected SNPs and he created protein models for the different combinations of SNPs. And for each of them, he calculated the binding affinity for a number of biologically relevant chemical compounds. For that, he used the Campbell database. And where JMOL in this case comes in, so all these almost 600,000 combinations of ligands and proteins is available on the web. This was something that would not be possible 20 years ago, but with all this growing list of open science tools, we can easily pick those bits of information. We can combine them. We can stand on the shoulders of the previous open science project and make something available in a much shorter time. And in this way, I think open science allows us to more quickly test our hypotheses. And this is just an example of it, but there are, I think, a lot of examples that show this. So what about interoperability? Here I want to return to OpenTox a bit. I mentioned the database already, but also that the Mbit software from ID Consult, the eNanoMapper software, if you like, that was implementing the reference standard of OpenTox that defined the application programming interface. And this programming interface was around computational toxicology or predictive toxicology. That had methods like what is the chemical structure of, what are the properties of that chemical, what is the biological toxicological prediction for that compound. So it had methods for interacting with the content of the database and to make toxicity predictions. And because this was all under an open application programming interface, I could actually extend BioClips, a piece of software that we were developing in Sweden, which was a chem/bioinformatics platform. I could integrate that API easily in the things that were being done there. For example, in the more general property prediction toolkit that Ulrich Spjut was developing in his PhD in the group where at the time at Uppsala, where I was a postdoc. And the API was clean. The API, the data exchange, was in different formats. I used the RDF representation there because I wanted to learn more about that, and that worked. And that allowed us to, without much funding, actually, to stand on their shoulders together with the other leg on the shoulder of BioClips and do something very efficiently. That is actually still a bit hard to do at this moment. We're actually trying to reproduce this now, extend that to other things, but we really need that API here. But BioClips is not existing anymore, so we're now using other tools to integrate these APIs. And the application here is read across and decision support. So if you're not into toxicology, basically it sort of comes down a bit to this. So if you have a new compound and you do not know anything about that compound, you're basically just going to look at other compounds, look around and read or translate the information that you have for that similar compound to your compound. So that is the read across idea. You look at multiple similar compounds and you combine that information, and based on that you take a decision. And that is what we can see here. And because of these open standards that were being used by the Open Talks project and various partners, so this was a global thing. There were multiple providers of such APIs. It could all be dynamically discovered. So what you see here in that decision support window there were actually computational services that were discovered on the web, whatever is online right now, as if it was sort of Twitter. Well, actually, that's not that unlikely. We actually, at some point, did it like this. We had computers talk to each other like humans talk to each other on Twitter, and they would ask for, do you have computational services that can predict something for a compound like this? You would get an answer, and in this way, do something that I think it was quite extraordinary. You can read the whole story in that chapter at the bottom there. BioClips integrated that, so because it was all machine actionable, you could do it not just for one compound, but you could do it for a lot of compounds and really do this read across, look at multiple compounds that are similar and see what toxicological data they had, allowing you to make a decision about your new unknown compound. But that needs a lot of standards. You need data exchange. I mentioned RDF already. Chemical Markup Language is an open standard that I've been playing with a lot. So in 2000, I mentioned the CDK that we started in 2000. And I was just a master's student at the time when we did that. And I got in contact with Chris Steinbeck and with Dan Gezelter actually at the time, mostly because they were making their material available in an open way. And the third and fourth, Peter Murray Rust and Henry Zappa did that with the Chemical Markup Language. And I was interested in Bioinformatics, and I saw those three things, and I realized that, well, if I combine those, I could use that for, a dictionary on organic chemistry. It doesn't exist anymore, but there was a website where I made knowledge about organic chemistry available on the web. And that came from a very practical reason. I was studying at the time organic chemistry, and I had to learn all these name reactions and all the trivial names of all these weird chemicals. And so I needed something like that could give me an overview of that information, and I could browse around hypertext like. And these three tools, JMOL, JChemPaint, and Chemical Markup Language together, that allowed me to put all this information on the web and share that with other people. So that is how I got in contact actually with these people. And this is how my journey in open science started. So this was in end of 97, 98 when this started to emerge, and the term open science was not actually even used at that moment. And the Woordenboek Organisch Dictionary on Organic Chemistry, it wasn't even using an open license because that whole concept was not really developed yet. That was coming in the next couple of years. But that Chemical Markup Language, that was really nice. It was XML, and a lot of people hate XML. They prefer YAML or JSON. But XML was actually nice because there was a large community around XML languages. And if you have one XML language, you can integrate that easily into other environments. And that is exactly what we did. So so this is back in the BioClips world in that period when I was in Sweden. Where we worked on BioClips and this prediction, quantitative structure activity relationship for toxicology, but not just for toxicology, also for drug discovery, and exchange of QSAR models, of these computational models to predict properties. That is also still something that is of high relevance to even the project that we're doing right now, like this virtual human platform that we're developing, where we want to talk or we want to collaborate about predictive models, but enable to talk about them, to review them, we need to be able to share them. So that's where this comes in. And we were using here XML languages, including the chemical markup language, to exchange this information. We could, in this way, really talk about how was the model created, what is it based on, what is the applicability domain, etc. On one of the latest slides, I should have another example of it, of how we were using a chemical markup language in dissemination and communication of knowledge. But first, a quick slide about the Blue Obelisk. So I mentioned JMOL, JCampaign, the CDK that resulted from that chemical markup language. But in the period between 2000 and 2010, where a lot of people were actually exploring this, how can we use open source, open standards, and open data? How can we use that in chemistry? And from this, the Blue Obelisk movement started, and we started talking about overlaps between different projects, common interest, common benefits, and yeah, we ended up with a good number of different projects. Some of them still used. Well, actually, many of them still used. Some of them highly cited. So you actually in the middle top there you see MBIT. So that is that database that was behind the OpenTox project. So that was one of the projects involved there. The use of standards to exchange information, that is still relevant. I said something about some positive things about journal articles, for example, that they're very sustainable because they're piece of paper or they're in PDF and they work now and they probably will work in 50 years from now too, if you still have access to it at that time. When it comes down to data and standards and source code, journal articles are not very suitable. But the whole field is still playing with this. You might have heard about the Journal of Open Source Software, for example, where the peer review is more about the software than about the article around it, and the article is kept short. And we see journals adopting open data standards, but we still haven't quite figured it out. So some people in Germany, well, and the community is growing, we actually say we should probably stop with the current concept of a journal article. And there is a lot to say for that, because if you look at this example, the fair chemical structures, and this is, yeah, it was proposed for the Journal of Bioinformatics. There is a second journal where that has adopted it right now. It proposed a very simple way of exchanging information. But the fact that this is a very basic approach and not everyone is using it is a good example of how far we are not yet, and how important a project like the NFDIs are. We have a long way to go, and it's going to take probably another 10 years before all the journals are actually adopting modern standards. I think the NFDIs have a very important role here in convincing the editorial boards and the publishers to do better than they are doing right now. And I'm happy that the Journal of Bioinformatics were listening to the authors, and we adopted that standard. Of course, the next thing is actually to make sure that other authors are actually using that, and that strong enforcing is not currently the case, I think. I will now go into a bit more away from the concepts, the solutions, and to how we have been using it. Actually, the difference is not that big as the previous slide suggested, but so some additional things from ongoing projects. One thing is the notebook science, the APIs, and the open risk net. So I mentioned BioClips integrating APIs, but the problem with that was that it was still all clicking. And BioClips, the second version of BioClips, it had a scripting environment, so that allowed you to replay an analysis, but it still basically started with someone clicking things. But if you if you want to be closer to how we do science, the notebook approach, recording exactly what you did so that you know when you do something for the next time, how that actually happens to be different from how you did it from the previous time, then notebooks are important. And I'm very happy that I had the chance at some point to meet Jean-Claude Bradley in person. He advocated for this concept of open notebook science very strongly. He actually has been with I don't know who was the president of the United States, but he has been actually advocating for open notebook science in the White House. And this over there is me handing me a Blue Obelisk award in Chicago at some point. I'm not sure if that was in 2012, probably actually. But this translation of the computation, which may contain bits that are manual, we see a lot of interesting development there as well. We have the open notebook science, that was the old thing, but we have more and more tools around that, like Jupyter notebooks or R Markdown or other ways of sharing how we actually perform the thing. And then the APIs are important. And what we see here in the bottom right here is the use of the Sparkle query language to automate how we get data from a database. And when it's a semantic, when it's a knowledge graph, of course, but within Jupyter Notebook, you can also extract data directly from data files and what's not. And in this way, more and more you can translate what you do, how you would do the science, into something that is written down. The open notebook idea, allowing other people to reproduce, to follow the steps that you did one by one. And in the top left, what we see there is a Jupyter notebook from the OpenRiskNet project, where Marvin Martens in our group worked together with Thomas Exener and other people in part of that a predictive toxicology, in this case using AOP, the adverse outcome pathway approach, which is a way of thinking about toxicology at a higher level to support the risk governance of the whole process. So it comes more from not from the scientific world, but more from the regulation perspective. But still linking to this high level of understanding the adverse outcomes and the key events that come before that, but with a strong link to the biological assays, the biological experiments that we do, all the way back to the molecular initiating event, which is even at a chemical level. And what Marvin Martens has been studying in his PhD study, his PhD work in our group, has been in how you can use ontologies and semantic web approaches to create a knowledge base to integrate all this information from the chemistry via the adverse outcomes to the biological data. And because all this is using open approaches, you can actually write this down as open notebooks, and that is something that we continue to use. So we're now translating these solutions from OpenRiskNet to this virtual human platform project that we are working on now. The APIs, they don't have to be semantic web. They don't have to be Sparkle. They can be REST APIs as well. And this is an interoperability tool, actually an Elixir recommended interoperability resource, BridgeDB, for identifying mapping. So different knowledge bases, different experimental data sets, they will use different identifiers to describe the chemical biological reality. And so you will need to make those resources interoperable by linking the right bits of information, which we do very often by means of identifiers, and a gene identifier, a data set identifier, or for you and me, a telephone number, for example. This is how we connect people. This is how we connect data. And so BridgeDB is an important tool for us, and here we're working with the European Open Science Cloud to further adoption of open standards, open solutions, to make this all this work easier to reuse. And well, actually last year we had a small grant from a national funder here to extend this. So you can actually run your open APIs here with your own identifying mapping data with Docker technology. And EOSC has been demoing this. So they have reproduced the BridgeDB API here on their platform. The data, the identifying mapping data as well, actually we use Wikidata for. So I haven't said much about Wikidata, but Wikidata is an awesome project. It's a sister project of Wikipedia. It's the machine readable version of Wikipedia. So in that sense, it's comparable to DBpedia that you may have heard about. And we're collecting a lot of information from across all domains here, and the domain where I'm more interested is chemistry here, and we use it now for identifying mapping between chemical structures. So these, what we see on this slide is a couple of examples there. In the top right, we see the hierarchy of chemical classes and specific chemicals there, specific chemical in orange, subclasses. So a bit of ontology you see reflected in Wikidata. It's cross-domain, so there is literature in there. So the idea of citation needed that you have in Wikipedia is very strongly embedded in Wikidata as well. So all facts there are ideally linked to literature that supports it, to references that support that, and that is widely adopted, and that allows us, for example, to give an overview of when did people actually do research about this chemical. Something that we will see later. And for each chemical, there's a list of databases that have more information about that chemical. So in this case, this linked data approach is a very important aspect of Wikidata. And that is both of those things are very useful in the interoperability and the reuse of data. So in the left bottom right there, we see different variations of acetic acid. With different isotopes or charged versus uncharged. And what we see there is that we have here 6 different forms of acetic acid, and they all have different identifiers throughout different databases there. Biologically, if you have a biological sample where you have a bit of acetic acid in there as a byproduct of one of the metabolic reactions, you only have one concept there. There is no difference between neutral acetic acid and the acetate ion. There's an equilibrium, and you will have different forms with different isotopes. There is some forms that are more abundant than others, but at an identifier level, they're all different. So you can see how this complicates how we integrate data. But you can also see here is how something like Wikidata, which is becoming an interoperability resource in itself, actually help us overcome these things and find a nice balance between chemical precision and biological precision, something that I'm quite fascinated about. Name drop here of Scolia, which is this graphical interface around Wikidata, which created these tables and histogram and graph that we see on this slide. Will not say much about this, but this is about this slide. But this reflects that charged versus uncharged thing, and we know how to capture this accurately. That we did, for example, a couple of 10 years ago already in the Open Facts project. So that's his scientific lenses paper there. We know how to do this, but we still need that information. Keeping track of literature, keeping track of knowledge, that is something that is really important for us. And this helps us actually not just capture past knowledge, but also to support research communities. And NanoCommons was doing this for the Nano Safety Cluster. The Nano Safety Cluster was this large group of projects in looking into the toxicology and the risks of nanomaterials, or the absence of that, ideally that actually safe by design paradigm. And then the access to literature, keeping updates to this literature is actually what are other people doing is a problem. You also know, well, you all know the flood of literature that we get every day now. So this notion that you want to have sort of a table of contents, but not at a journal level, but at a chemical level. This is something that we have been playing with in NanoCommons together with the Scolia project, and that is what we see here. By giving nanomaterials a unique identifier, and we can use those identifiers in articles. And in this case, it's the Joint Research Center nanomaterial identifiers that are increasingly used in research papers. And by using those identifiers, we can more easily integrate the both. And that results in that bottom histogram there that shows the number of articles about JRC materials over time. And we're using Wikidata there again, and the eNanoMapper ontology. And by having all this information machine readable, we can actually just create an RSS feed, a news feed, a rich site summary feed, so people can just follow that feed and get an automatic alert when a new article becomes available about one of the materials they're interested in. So interoperability to support ongoing research, not just for knowledge lookup. So yeah, regarding that reuse, that standing on the shoulders, how does that translate to projects? So this is an overview of the projects, and I realized actually NanoCommons finished at the end of last year, so that project is over. FAIR Plus just finished a short time ago as well. Not all the papers are out yet, but this is an overview basically of the projects that I'm involved in right now, or trying to. Some a bit more than others, but it's quite stressful with so many projects, I have to say. But it works because they actually reuse a lot of these open approaches. Every project does not have to reinvent the wheels, but they can still stand on the shoulders. There is an overlap in the solutions that they use, so that they can focus on the application of that solution for the deliverables in that project. All of these projects, they have their own unique bits, quite a bit of that actually, but there is still a good bit of overlap as well. I honestly do not see how I could have done this without open science. And likewise, I don't think any of these projects would be as successful without open science either, without this overlap, without this common interest. So the PHP for Safety project, that is a project that started almost 1.5 years ago now. We're approaching the 2 year. We just had a third designathon meeting, and one of the deliverables that I'm involved here is actually reproducing that AOP link Jupyter notebook that I mentioned earlier. So what we need to do for that is making sure that the services that were integrated in that notebook are still available. And to do so, we're trying to reuse the standard protocols that OpenWisNet supported. Also because other projects like NanoCommons and NanoSolvit are doing this as well. So to ensure that you sustain that interoperability, commitment to open standards is particularly important. Now for HTTP, this is pretty obvious. For other things, it's less obvious because it is not as established yet. HTTP is probably not even a question, but Sparkle, for example, or RDF or Bioschemas, those are still emerging. Those are still where you can have a discussion about, is that the right approach? So by using all these tools, we're creating here a virtual human, that's VHP, virtual human platform for safety assessment, and we're integrating all these sources so that we can actually do a computational prediction of the safety of a compound. But unlike what I showed earlier with BioClips, where we predict individual biological endpoints here we need to integrate basically everything about the virtual human. So we need to know how are we exposed to the chemicals? So what are the working conditions? How does it dissolve? How does the potential chemical compound distribute over the human body? How does it interact with the tissues that it passes? A lot of information that requires a lot of integration. And we had the ambitious idea that we might be able to do it. And with 2 years into the project, we're making progress, but we're also realizing that we have a good bit of work to be done. But again, because we do everything open science, we also have this notion that even if we don't get there all the way, a lot of the building blocks will be sustained beyond this project. They will be continue to be used in other projects. So how do we actually maintain that collaboration between projects? And here the Elixir is doing this for a lot of life sciences fields. So Elixir is this large organization to support the sustainability of the life sciences solutions in life sciences. And within that context, for toxicology, we have created proposed the toxicology community that led to the white paper that we see on the right side there. We were accepted last year somewhere, and we're now setting up the first funded activities. But yeah, we actually started in 2018 already, I think, and there have been we started that collaboration between people doing toxicology there already, along with quite a few different projects, and some of the results you saw already. And this is important. And I'm now that I say this out loud, I'm not actually sure why I'm telling this, because as NFDI, as part of this large or huge community, you know all about what it is to set up a community like this. And so Elixir has been doing that for us for the predictive nanosafety, and this is a slight overview of that. Some more information. So if there's anyone here that is doing something more towards toxicology, please do feel free to join this community and work together on this this collaboration, this reuse via open standards, open solutions, fair solutions, etc. I'm not entirely sure what we decided in the end on the time. I have a few more slides left. I'm approaching the end. I'm happy that so many people are still here. I just want to say a few things that I think are quite of interest to all the NFDIs, and one of that is Bioschemas. And the reason for that is it is some sort of a light knowledge graph approach. It is semantic, but it captures a bit of information. It captures a bit more information than you would do if you deposit data in a repository, for example. It would add a bit more information. Bioschemas is a project there under the umbrella of Elixir, actually, that tries to capture a bit more information than just the title of a data set or the title of a database, and go towards indexing the content a bit more. So from this context of the toxicology community, we've contributed to Molecular Entity that was started, I think it was started by Ricardo and David from the Campbell team at the time in 2018. I have a question mark there. I got introduced to it via them. So that was my first introduction. And from the nanosafety fields, we introduced a chemical substance. And in doing so, we can annotate pages of information that are about a chemical entity, molecular entity, or a chemical substance. And Bioschemas is actively used by the NFDI for chemistry, actually. So there are some examples of adoption there in the German NFDI community. And the nice thing there is that it allows search engines to pick it up, and we're working on that. It works well for data sets because there Google actually indexes these data sets. Google, as far as we know, does not index things at a molecular entity or a chemical substance level yet, but in the nanosafety field, we are doing this. And This is an example of a page actually where we're communicating open data sets in the field of nano safety. Started in the NanoCommons project. We're continuing this from the nano safety cluster field yet, but here we have annotation in Bioschemas, and that allows that these data sets are found in the Google dataset search engine. And because it's Bioschemas, we can annotate it more precisely with what is the chemical substance or the molecular entity that this data set is about, or what are the chemical entities described in this data set. So that deeper indexing, that more interesting indexing, that to encourage the actual reuse, to make it findable by asking, give me all data about this chemical. That is enabled by Bioschemas. I mentioned identifiers earlier, and that is a problem that we run into. You do not always have identifiers, so sometimes you need to create your new identifier. If you create a new database, this comes almost automatically. It takes a bit of effort to make it global, to make it unique in sorts, and this is what we did for nanomaterials. And if you see at the title of this article by Jean-Vianne in our group, the undisclosed there, and this is something that we observed in our community. Not everyone was ready for open science yet, for quite a few different reasons. This is in the RISCOM project. One of our partners is writing this up now. The various aspects of data sharing. Well, not unique, but she's looking at it from an ethical and moral perspective, or in a frameworks from that area. But we needed that, and we still needed every project to actually annotate their project deliverables, the PDFs, the reports at the end, along with the data, with unique identifiers, so that we can accurately link those narratives to the data that is shared as well. And it will become available at some point, sometimes 1 or more years after the end of the project. But you need to start using the identifiers now, because recovering what goes together actually after a couple of years is pretty hard in a project with multiple projects at different institutes, very often where the original researchers moved on to another research position somewhere totally different. And as an example here, here we have 6 different nanomaterials, but they actually have 60 different synonyms. But there are actually just 6 different materials. And by using this identifier, anyone can use whatever synonym is most suitable for the narrative at whatever moment. But by stating the identifier, we know exactly what that narrative goes with. We set it up. It's a bit of an experiment, because using identifiers is not something that comes easily. You need a database, but if there is no data yet available, which is something that we had, there were no physical chemical characterization of the nanomaterials yet. So what chemical are we exactly specifically talking about? Does the material change when you purify it? All those kind of things, something that you can express when you use these identifiers. And we're using here a semantic web approach to register these materials. And because it's RDF, because it's semantic web, we can grow this network easily in a distributed way, in a semantic way, without the necessary need for a single person. So really an open science approach to this problem of getting data together over a longer time. I promised to say a bit about WikiPathways in the abstract, so I will do that. This is our new website. I do not know how many people here are into biological pathways, but I also show it here actually to show that the diversity of of knowledge and data that we have. And in this case, we have diagrams that we want to make machine actionable, that we want to make machine readable. And WikiPathways is doing that for biological processes. And we just underwent a transition from an old code base to something new based on GitHub actions that makes us, it's more open, it uses newer, more modern open standards, and that allows us to further grow. And the kind of machine readable that you can think about is a description of these biological processes at a chemical level. And I hope, and it should probably not take a lot of imagination, how these processes, how that translate to chemistry and physics, where you also have a lot of processes that you might want to describe. Organic synthesis is a very clear example, very close to biological enzymatic reactions, for example. But the point here is that you can apply ontologies at an arbitrary level in quite a few different ways. We know how to do that from the chem/bioinformatics world. A lot of opportunity there, and this too is semantic web. So we have Sparkle query language that we use, and I dropped that here because this is very powerful, because we have multiple resources in the life sciences that expose their data as semantic web with ontologies, and that makes it so much easier to integrate data, that we can define our open notebooks as a Sparkle query as here. And if you look at that line 14, so this is a query that we run against the WikiPathways knowledge base, but in line 14, part of that query is against the Nextprot knowledge base, which is running in Switzerland. And whereas ours is running in Maastricht, actually. The semantic WikiPathways is running in our group. We can do this integration and repeat that thing. Now, yeah, a Sparkle query is quite abstract, is quite technical, but we can embed that query. We can explain the narrative again in a Jupyter notebook, for example, where we explain queries like this step by step, taking the advantage of all the open solutions that we have nowadays. That gets me to the conclusion. So we started with open is not fair, is not necessarily used. We looked at various aspects of that. The sustainability, the reuse. I hope these examples show a bit how we manage to grow our vocabulary of open approaches to such a level that we can really tackle the latest scientific questions that we have. And there's what I didn't show, a nice example here is the how we use various approaches to understand the SARS-CoV-2 virus, what this is biologically doing, and only by using these open approaches, we were able to get more than I think something like 150 researchers together that were really looking into the biological details of how that new virus worked, allowing us to create maps that describe that information. But for that, you need open, you need actual reuse. So you need to enable that. You do that with open licenses. You do that with open standards so that other people can actually integrate the information. The interoperability is important, but as always, you also need the social aspects to get people together to really make it reused. And that is something that we're now looking into, because that is actually less obvious than we thought. And part of that is because how we fund research does not always reward these kind of approaches. Sometimes it actually rewards us doing something not open, not fair. So something to think about. So what I showed, the open approaches for identifiers, the use of ontologies here, semantic web, the RDF, other standards, REST APIs in general, all different ways to make sure that what we do, that we have control about the scientific quality of all the research that we do. I hope this presentation gives you some idea of how we have been doing that in biochemistry. That gets me basically to the last slide, except for the two following things. I showed a lot of research. I've collaborated with a lot of people. This is a long list of, and it's not completely complete, but it's based on co-authors that I've been writing research papers with, and and the group that I'm working now is the BiGCaT, the Bioinformatics group. Professor Chris Evelo, that's our chair. Suzanne is associate professor, and Lars, Freddie, and me are assistant professors. And we have a very nice group of people that are all into open science and really wanting to push these things forward because they see how important this collaboration is. With that, I'd like to thank everyone for your attention and welcome your questions.
Thank you so much for such an inspiring presentation. The truth is that many of the tools you showed, I wish we could offer to our community at some point. Is there any questions from the audience? Please feel free. You can either write them down in the Q&A, or you can also raise your hand if you want to ask it in person. And while we wait for some questions, I have one of my own. You have worked in a lot of different projects regarding open science and the sharing of data. And I was wondering, what is the biggest challenge, in your opinion, to get people to share their data?
I would say in my feeling at this moment, the two categories of main problems here. One is my data is not ready. That is one aspect. So uncertainty about the quality of the data. They realize we need more experiments, for example, or the experiment just failed. The data is not good enough, even if it's published, because people do experience this publication pressure. So that can be a reason. They have to publish articles because that's what they're supposed to do, but the data did not perhaps have enough peer review as they would have liked. The other thing is that data still is seen as competitive. When I started my PhD, it was not uncommon to hear statements like, "Well, if you want to have access to my data, I want to be involved in that. First of all, because I want to make sure that you use the data correctly." That is one aspect of that overlaps with that first point that I mentioned. The other thing is that, well, you can have the data, but I want to be author on that follow-up paper. So again, that publication pressure kind of thing. But also translating that second one to this data that ensures me that I will be able to do research next year as well. That data is part of my power to get the next grant funding. So that kind of idea. I think these things are not helping. For the industry, you see. I think similar things here. But I do like to stress this is definitely not just for industry. At an academic level, these things are definitely real world too.
And nowadays, when we are doing research proposals, you have now these research data management plans that are being asked. Do you think that that's enough to really move to this transition to open science, or do you think that that's still work to be done?
Yeah, that's a very good question. So in the PHP project, we have someone doing research into this. We have to write these research data management plans. We understand why we do it, but I do not have the impression that they are working yet. I think at this moment they are not working. And part of that is that a lot of the tools that we have around research data management plans are too focused at the F and the A, at the findable and the accessible. But FAIR is not in black and white. It's a direction that you have to go, and to actually actually really trigger real reuse, that is a lot more expensive than what you typically can write down in a data management plan, which is very often just focusing on things like where can you find that data again 5 years after the end of the project. That is not about reuse. That is more about accountability, perhaps. So different objectives of FAIR. So that was my point also about we should not get ourselves too distracted from meeting FAIR requirements, but really focus on the interoperability and the reuse. I think that's a lot more interesting. The other thing is that I think, and I definitely underestimated that myself as well, because I have been doing data analysis for so long. I know where I want to go. But generally, when we write a data management plan at the start of the project, we have no idea yet what experiments we're going to do. We don't know how much data will come out of that. Sometimes we still have to buy the equipment to run the experiments, and God knows what data formats that will spit out those platforms. So writing a data management plan at the start of the project doesn't work. And so this you see reflected in ideas that data management plan has to be a live document so that you update it all the time. But if you do not make enough time and resources available for that, it becomes an administrative burden and not something that benefits the eventual interoperability and reusability.
Thank you so much. Yes, I agree with you. Another question that I have is NFDI for CAT is starting to work now or starting in this open science road.
Mm-hmm.
And if you could give us an advice from your years of experience, what would be the most important topics where we should really focus on now that we are just starting?
Read The Cathedral and the Bazaar. That was a write-up from a long time ago, I think probably something like 25 years ago, The Cathedral and the Bazaar. And this is a nice example of a nice description. It was oriented at source code, but it very nicely translates to doing research as well. And one of the ideas there was the release soon, release often that I mentioned there. In a cathedral, you would first publish the Nature article probably 2 or 3 years after the end of the project. So probably 4 or 5 years after you start your research. That is 5% of your lifetime, if you're lucky, that waiting period. If we really want to solve the problems that we're facing, I think as a society right now, we need to be quicker than that. So my advice would be wherever you can, and it's definitely not an all or nothing, but where you can do release soon, release often. Look at the bits where you can share it because it's less important. It can be, for example, an open standard that is not essential to your research, that will not jeopardize your Nature or Science article or whatever your preferred journal is. In that sense, I'd rather see 100% of the researchers doing 10% open science than 10% of the people doing 100% open science. So that is the second thing. So where you can look for that mutual benefit as soon as possible, because it takes a bit of time to build up. But if you start sharing things now, you will start collaboration with people now instead of 5 years from now. I think my research career would have looked at least quite different if it would have existed at all. If I had not run into Peter Marust at Cambridge University, Chris Steinbeck in Germany, and Dan Gezelter in South Bend 25 years ago, I would not have been here where I would now. I would have not been invited to come to do part of my PhD at Cambridge University because they would not have known about me. open science as a way to create collaboration. And you can always collaborate on something. It doesn't have to be your crown jewels. So release soon, release often, and focus on a bit of open science. Start with a bit of open science, and not try to do everything open science necessarily. You don't have to.
That's really nice advice, really nice words. Another question. You talk about the journals changing the way they publish. Could you maybe, I don't know, explain a little more or elaborate a little more how this change should maybe look or start? Like, should people, because now you are not required, but you are asked to upload your data and the metadata and stuff like that, but that's probably not enough. So
no. So
change start. Yeah.
Yeah, so some journals implement their data standard in different ways. So a lot of journals they're currently still happy with statements like data available upon request. Now people have been studying that, and the meaning of that that phrase is actually pretty dark. Less than 10% of the situations you would actually get access to the data if you kindly requested it. For people have been looking into the reasons why people would not give data there, with sometimes a very weird reason, like, oh, they didn't ask nicely enough, that kind of stuff, or things like, oh, but actually we actually don't have the data anymore, or I can't find it, that kind of thing. So they by complying to journal standards, it doesn't necessarily mean that you can really reuse it. The other thing is that what we also see is that people make things downloadable, but well, we see really weird things there. It can be binary blobs of source code. It can be data in some obscure format, or data without sufficient metadata. So you have access to it, but it's totally not interoperable and not reusable. And so if we focus too much on it is there, then we're missing the point, basically. So currently journals are trying to do something, but it is they have not kept up with open science in general, I think. So open science has been around now for at least 20 years, and the journals they're still thinking in terms of what was possible 20 years ago.
Thank you so much. One last question for my part. This question is for me. You talk about some projects that you're currently working on, for example, Bioschemas and your own work in Wikidata. I think there's a lot of synergies with what we want to do, and it would be nice to maybe work together or get together working on these topics or within these projects. How could we go about it? So emails, is there, I don't know, some kind of events that you're organizing where we could attend, or how could we get in touch on that matter?
Yeah. Yeah, so one of the nice things of doing open sciences is that this communication is typically open as well. The collaboration, of course, is a bit complicated with the need for funding nowadays. Few people nowadays still have some time left at the end of the week. Everyone is so overbooked, so pre-scheduled already with the grants that starting a new collaboration can be a bit tricky. My advice here is to actually talk about it openly, because if you ask someone specifically, if you would ask me, for example, then I would have to say, actually, probably we should talk again next November, because that's the first time where I hope to have a bit of free time again. It won't be before that. That doesn't mean that I can't squeeze in here and there a bit more information, a bit more detail about what I've been doing. And this presentation gave an overview, for example, but you can reach me via Mastodon. My Mastodon account there is on this slide. And by having that communication openly, you give people the chance actually, oh, but wait, we're actually doing something really interesting. And this sometimes can be a bit frustrating because I'm sort of expected to bring in a lot of money from my university. So in that sense, I want to be involved in every project. Yeah, give me the money, but realistically, I don't have the time, and so it can be painful, but it at the other hand. It is very important that these things, these collaborations start happening or are happening, and a lot of them are happening. So do it openly. Some strategic thing is necessary. NFDI funding is for Germany, not outside that. I won't be able to get NFDI funding, for example, and vice versa. So this is the case. So look at the mutual benefit and collaborate with whoever wants to collaborate, and open discussions. I mean, for me, it started with an email.
Thank you so much. And one last thing, is there a similar initiative like NFDI for the Netherlands?
I wish. Well, there are some organizations, but not at this scale. There is definitely. I mean, we have the eScience Center. That is, but that is set up differently, and it's a separate institute. But they collaborate with a lot of research institutes. We have our own supercomputing platform that serve, for example, but something like NFDI for the life sciences, we had the Dutch Technologies DTL, Dutch Technology Center for the Life Sciences. We have Health-RI. That is the next thing. That is for for health research infrastructures. But it is still a bit isolated. The idea, I mean, you have subsections of NFDI, but this idea of NFDI and the amount of funding behind that, we don't have that equivalent. So that was a bit frustrating when that news came about, because you can do fair, and we already see that in some fields where we in the Netherlands, we cannot compete with that level of fairness with what is happening in Germany right now. So unless NFDI they start becoming very open science, then we will benefit from it as well, of course. Hint.
Thank you so much. There is no more questions coming in. I don't know if, please, if there's a question now.
If other questions come up later, just post them on Mastodon. I'm checking that regularly. Send me a question there. Would be most welcome.
Thank you so much. Now I know also where to reach you. Mastodon. Thank you so much for taking the time for this presentation. Thank you for the invitation and the opportunity. I will send you the link with our YouTube video, because this talk will be uploaded to YouTube. And yeah, I'm looking forward seeing you again in person, hopefully soon. Yeah, otherwise, thank you very much and have a nice day. Thank you, everyone. Have a nice day too. Bye.